It's time for enzymes, which means it's time for awesome. Enzymes are awesome. They're these little proteins in your cell that make chemical reactions happen. Not only make them happen right. Now, there are thousands of chemical reactions that can happen inside of cells, and your body needs them to happen just so. We use a lot of symbols for this, at least in biology, because showing big chemical formulas up there is just too confusing. So it's easier to drive the point home saying we got a square chemical and a triangle chemical. We want to combine those and make a little house out of it. Now, chemistry in a beaker, when you just add chemicals together, a lot of stuff could happen. You could get any of these products. Enzymes make sure that you don't get any of those, just the stuff you want. In my book, that's awesome. All right, so enzymes basically help chemical reactions out. They make them happen faster, better, and more accurate than the chemical reaction would be on its own. You know, not all analogies are perfect, but this one is pretty close. I think of enzymes like little specialized tools, whether it's in the garage or the kitchen. Enzymes are not general tools like knives and spoons. These things are fine and they're great for what they do. They can do all kinds of things, but not quite as specialized as enzymes make it. Enzymes are more like that carrot peeler that makes it just so easy. I mean, you could use a knife to do that, but it'd be so difficult. You could use a knife to chop up garlic, but isn't it so much easier with a garlic press? Now, what else can you do with a garlic press? Really not much. If you think of something, put it in the comments, but I only press garlic with mine. It does a fantastic job, but it does just one thing. And enzymes are a lot like that. They're like can openers. I mean, what else are you going to do with a can opener? You know, throw it at somebody if they're attacking you? Probably not that effective. It opens cans, and that's all it does, but it's so much more effective than anything else. You know, you got things that do such specialized tasks. You ever seen one of these? This is an old tool. Spins around. That's like one of the most specialized tools of all. Stick around to the end. I'll tell you what it is. It's kind of a weird one. But enzymes are like these specialized tools in a few ways. One, you can reuse them just again and again. You can keep pressing garlic clove after garlic clove with that. Same with enzymes. Two, they make the job easier and faster. Three, they make sure that you get the right product, right? Yeah, you could just smash a can with a hammer, but the can opener makes sure that it gets open nice and neatly. So like enzymes, the tools don't do the job for us, and they don't power the reaction, but they sure make it a whole lot easier. A couple bits about just chemical reactions in general, whether they're catalyzed by an enzyme or not. One, they're not spontaneous, and two, you can get a variety of products out of them. And like I said before, that's why we use enzymes. That controls that factor. But let's talk about the spontaneous thing. Now, this book of matches has been sitting in my kitchen drawer for several years now. And you know what? None of them have ever burst into flames on their own. That's what I mean by not spontaneous. If you just had those two chemicals, well, shapes, in a solution, they wouldn't necessarily react with each other. Now, maybe if you added a bunch of heat or light energy or something like that, then they might combine, but probably not. It's not spontaneous. They have to get started. They need to be guided in some way. And again, if they're in a beaker, you might get all kinds of crazy products, not just the one you want. So when we want this match to burst into flames, we add friction, and that starts it. Well, that was more exciting than the next part. We're going to graph it because we're scientists. That's what we do. We graph things. So here we go. Kind of standard. Looking at time, this is the start of the reaction, end of the reaction. So beginning and end. There we go. Then we're graphing the amount of energy. So let's take these two things here. Now, if we were to say how much energy do they have compared to this one, probably less. So we have these at the beginning of the reaction. I'll put them over here. And on a graph, it would look something like that. Now at the end of the reaction, I'm going to combine things. Usually that adds energy to it. It requires energy, stores energy in those chemical bonds. So that is going to have more energy. All right, so when I do this reaction, that means one, I'm going to need to put energy into it. And two, it's not going to happen spontaneously. Remember, most chemical reactions are reversible. Let me come back to that one. When I add energy, I'm going to have to add a bunch, and then it settles back down into the amount of energy that that molecule contains. So I actually need to sort of overshoot and add more energy. So this is known as the energy of activation. Kind of makes sense, actually. It's an intuitive term. We can symbolize that this way. Energy of activation. The amount of energy from here to here that I needed to put in to get this reaction to happen. Well, one thing enzymes do is they reduce that and make it easier. So this makes it more possible in our body without adding giant amounts of energy. A lot more efficient, kind of like those tools. I said a lot of reactions are reversible. So what's keeping this from going back to this? Well, this barrier. I would still have to actually add energy to this to break it down. Okay, so that sort of extra energy actually keeps the molecule stable. That's the same reason that those matches don't just spontaneously burst into flames. Let's actually look at an exergonic reaction, the opposite, where we start with a lot of energy and we end up with less, more like the matches. It's kind of like lighting those matches. I had a lot of energy stored in the match itself, and then when it got broken down to smoke and carbon dioxide, water, all the stuff that was given off chemically, what I ended up with, all those chemicals and the ash, have less energy. They're broken down. So that kind of makes sense in terms of this. A lot of energy was given off in that reaction. Right? That's the exergonic part. And in our bodies, we would probably try to capture that energy somewhere else. But that was just a match. Now, enzymes are kind of like the head of those matches. Now, matches made of wood or cardboard, right? And I could get that stuff to light on fire just by adding a lot of friction. But that would take a long time. That would be really hard, kind of like rubbing two sticks together. So the tip of the matches is kind of like an enzyme-assisted reaction. There, I just have to gently strike it. I don't have to put lots and lots of effort into it to get it to start reacting on its own and giving off energy so that it can keep going and burning the next bit of cardboard wood. In that sense, the matches are a little like an enzyme-mediated or enzyme-facilitated reaction. They're a little easier than they normally would be. Key part of enzymes, they make things easier, like those little specialty tools. All right, so enzymes make things easier. They guide us to chemical products. What does that look like? All right, so hopefully I've built them up a lot. You're really excited to understand how do enzymes work. Here we go. About the only thing scientists like more than a good graph is a good diagram. This is probably about the simplest and honestly kind of lamest diagram we'll see about how enzymes work. So let's translate this into a little more pictorial diagram, give us a better sense of what's going on. So we start with this enzyme. It's a big protein, usually, and a substrate. So that would be the reactant. Whatever chemical the enzyme is acting on, usually it's much, much smaller. So we have enzyme and substrate separate, and then something happens. That's what the arrow always means. And now we have enzyme and substrate together, the enzyme-substrate complex here. And this substrate is always specifically suited to fit right in this part of the enzyme. That part is a crucial part of the enzyme. That's called the active site. So the active site is where the substrate fits. That facilitates a chemical reaction, kind of gets it in this little nook, is able to push and pull on some of the chemical bonds, isolate it from the rest of the environment. That's what makes the reaction easier to happen. And so the chemical reaction happens, enzyme is released, and then we have our products, whatever the enzyme produces. And enzymes can put molecules together, it can tear molecules apart. They vary. Obviously in the digestive system, most of the enzymes there are breaking molecules apart. But they can also build things up, like the proteins that make up hair and muscle. Enzymes do that too. Okay, so there's a basic diagram. One other key piece I want you to notice about this is that the enzyme came out unchanged. And so one other thing you might add to this diagram is the idea that that enzyme gets reused. It's an important concept. If you had to make an enzyme for every molecule that you were going to digest, well, you'd probably spend more energy making the enzymes than you would getting out of the food that you digested. So the fact that they can be reused means we don't need to make a lot of them, and they just keep doing their job over and over and over again. Well, while this somewhat illustrates what an enzyme reaction is like, I think in a cell it looks a little bit different. There's more than just one substrate molecule, etc. Take a look at this animation and see if it brings things a little more to life. So although not a perfect illustration, this is a little more what I think of when I think about enzymes. So here's our enzyme in here, really nice big protein, and these guys out here are our substrates. Now those substrate molecules and the enzyme are always in motion, right? Just everything is in motion. But when one of these substrate molecules lands in that active site of the enzyme, okay, so not all motion stops, but when it lands there, then a reaction can take place. In this case, that enzyme breaks it down into two other molecules, which it then releases. Everything's still moving around, and let's say another substrate molecule crashes into place, converts, releases another one, and this just keeps going and going. Notice that the enzyme remains unchanged. It can just keep doing this all day long. It's like Captain America. Well, I don't know if I've convinced you that enzymes are awesome, but I really think that life would not be possible without them. So they're so necessary because life in the cell is a bunch of chemical reactions. And those chemical reactions need to happen just right in order to support life. Remember, enzymes are like little tools. And I told you earlier, I'd tell you what this is. This is a pie crimper and sealer. So you run it around the edge of the pie and it creates those little ridges and seals it off. Quirky little tools. Whoever thought of those probably thought of inventing enzymes too. Well, that's it for this time. Until next time, remember, life is awesome.